Thank you, everybody, for joining this meeting. This is the last one before lunchtime. So we are already 2 minutes late, so we should get started. I'm Tim Turkis. I'm a radiologist at Indiana University.
I'm Hanno Steen, Boston Children's Hospital and Harvard Medical School, the co-chair of this session. So we are really looking forward to learn more about pushing the frontiers of pancreatology using digital AI-driven tools and wide screens.
And our first speaker is Nikhil Thuruvengadam. He will talk about embracing open source data mining in pancreatology.
Good morning, everyone. I just wanted to thank the organizers of APA and the moderators for having me talk. Today I'm going to be talking about embracing open source data mining in pancreatology. I don't have any disclosures relevant to this talk. So getting into the outline for this talk, we'll first talk about definitions, a framework for open source data mining, and then examples of successful data mining in pancreatology. Just getting into definitions so we can all talk about the same thing. When we talk about open source code or technology, the idea is it's a decentralized model of software development where the source code is publicly accessible for anyone to use, modify, and distribute. Typically means the software is stored in a repository with a common one being GitHub, and multiple people can modify it at the same time. When we talk about open data, this is information that's freely available for anyone to access, use, and share for any purpose without restrictions. Usually there's a requirement to attribute the data to a source, but you can use it however you want to. The last definition is data mining, and this is a process of discovering patterns, correlations, and trends in large data sets to transform raw data into actionable information. It usually involves complex statistical methods of matching and making comparisons, as well as machine learning techniques. When we talk about sources for data mining, it really started more on the basic science side, with the development of different omics data sets, starting with genomic information, metabolites, proteins, even more recently radiology. And it was really the advent of both payers and journals requiring this information be deposited into registries and data sets that led to kind of more open omics data. On the clinical side, open public data sets have really been around for the last 30, 40 years, and it started with both federal and state governments, sampling different populations with things like NHANES and AdHealth, and with national survey studies, and then making the data available for researchers to use. When we talk about closed public and private data sets, this is more of a newer kind of innovation, but it started with administrative claims data sets, added lab and pharmacy data, and more recently is either EHR enriched or based in the electronic health record itself. This is often proprietary, requires data use agreements, may have significant licensing costs to the investigators, but contains a wealth of information. And the last source, and this is, in the last decade, has really been social media-based data sets using X, TikTok, and Meta, where for health researchers, they provide with licensing data sets on a wealth of information that's being shared on social media. When we look at challenges in medical data mining, you can see in this figure on the right, there are several challenges, but the biggest challenge to date has been really disparate data sets. Some data sets will store their data horizontally, meaning each patient is in one row. Others, one patient will be in a column. They may use different uniformity in dates. For example, some data sets may subtract the days from January 1st, 1960. Others may use another reference date altogether. Coding can be all over the place. They can use ICD-10 procedure codes or CPT codes, and this lack of uniformity has made it challenging. Another big challenge is really the duplication of data. We assume that each patient in a data set is unique, but when combining multiple data sets, one patient may have contributed follow-up at multiple data sets. When you're talking about the aggregate, which is the hundreds of millions of patients, it's not that big of a challenge, but when you start getting in more specific conditions that are not as common, a small number of patients may contribute a lot of the follow-up time. The last and kind of most significant challenge has really been the lack of availability of paper records. A lot of hospital systems moved to an electronic health record in the last decade or two. And for example, if a patient was diagnosed with type 2 diabetes, say in 2007, but it was on a paper record, and then you have the first electronic visit in 2014 for diabetes, that may be misclassified as a new diagnosis when they always had diabetes that entire time. Getting into frameworks for open source data mining, there really is a consortium called the Observational Health Data Sciences and Informatics Program based at Columbia University. That's a multi-institution, multi-stakeholder consortium that really wanted to create a platform for open source analytics with big health data. In this figure, you kind of see what their goal or schema is. The idea is you take source data either from a data warehouse, from the electronic health record, or other sources of patient-level data. You use a tool to transform it to a standardized, de-identified patient data set using a common data model, perform large-scale analytics, and then report the results in a standardized format in their data repository. In order to achieve this, we really need a protocol for standardizing data. It starts with the data structure itself, right? How do we want to structure the table? How do we want to structure the names of fields, the types of fields? It goes to the content. We need a standard vocabulary to codify clinical domains. How are we going to code procedures, diagnoses? It then moves to how we define cohorts and covariates. For example, smoking can be defined in a simple way as passive, a never smoker, current smoker, former smoker, or with a lot more granularity. But a common definition really helps us use the same tool across data sets. It then proceeds to how do we report analysis and results. If we do that in a standardized way, it's much easier to combine the data and compare them. A real leap forward in this regard is the OMOP Common Data Model. And this came out of OHDSI and a lot of other stakeholders that created a model for both administrative health claims and electronic health records. The idea is it could capture unique fields from both inpatient and outpatient settings, include things like cost data, procedures, medications, and enable the standardization of structure, content, and analytics. This kind of gives you an example of how we would use this common data model. At the bottom, you can see the source codes in the data set. They could be ICD-10 codes, they can even be directly from omics data or from SNOMED. And then the idea is you map them using a vocabulary to SNOMED CT codes, which you can then use across data sets. And you can do a similar process for medications and for adverse events using common terminology like MedDRA across data sets. When we look at kind of the progress of using a common data model, this is a table that just illustrates some of the possibilities. The All of Us research cohort from the beginning has used this common data model, really facilitating an analytics platform. A lot of administrative claims data sets, Optum, MarketScan, and TriNetX have adopted this as well, making cross-database analytics possible, as well as multiple federal and institutional data sets. When we get to this roadmap that OHDSI provided, this is kind of what they envisioned, and some of this is starting to happen, and a lot of this is kind of a future direction. But the idea is once you have in the OMAP data format, they provide tools to profile the database, to see if you can define the cohort, is it even feasible to assess these outcomes with tools like Calypso, and then they provide specific tools like Plato and Homer that allow you to do population-level analytics. Next, I'll get into some examples of data mining in pancreatology with the caveat that this field is really in its infancy, so you're just starting to see what is potentially possible, but it really scratches the surface. So the first example I'll get into is a study that aimed to use machine learning to predict exocrine pancreatic insufficiency using Truman MarketScan. They specifically looked at patients with pancreatic disorders, malabsorption syndromes, and HIV, and excluded pancreatic cancer and prior pancreatic resection. They were using an open source Python machine learning model to classify patients as actual positive EPI, actual negative, because they'd been tested, or undiagnosed EPI. They used both a training and testing set where clinicians manually label these patients in these categories. And as you can see, the area under the curve was excellent for both the training and testing data sets. What you can see in this figure, it's a little small, is that the model was able to say if someone was likely or highly likely to have EPI, and then link it to EHR flags to allow patients to potentially be tested as being high risk for undiagnosed EPI. Then getting into a pharmacoepidemiology study, this is a study that we conducted using Optum Informatics. The question here is we were trying to understand what the relationship between regular statin usage in patients with acute pancreatitis and the outcome of interest being post-pancreatitis diabetes, defined by a combination of pharmacy and ICD codes. The nice thing here is we were able to use an open source cohort and statistical analytical tool that I'll introduce to you called Cohort Method. This is an analytical tool that was first developed in R, And basically, it allows you to create a cohort of new users and analyze different exposures with one command, as long as the data uses the OMOP common data model. And it really harnesses the power of Common Dictionary to be able to perform these analytics across data sets. We looked specifically in Optum, and what we found was that incidence of pancreatitis was lower in statin users compared to non-users. You can see this in this cumulative incidence curve going out to 12 years. And one strength of this approach is that we can repeat the analysis every year that Optum adds follow-up data, so the cohort grows over time and adds follow-up to see if this association remains. When we used a Cox proportional hazards model, we saw a significant reduction in post-pancreatitis diabetes with regular statin usage, and the findings were similar when we included potentially undiagnosed diabetes in the first 3 months. So the next thing I want to highlight is, a group published this in JAMA, and it's a really nice illustration of taking that approach but magnifying the scale of it. So they took 32 phase 3 RCTs when they were registered in clinicaltrials.gov, so they started enrollment but hadn't reported results. And they asked the question, can we replicate this using large-scale analytics in clinical data sets? They used both Optum and MarketScan for this, and they registered separately their protocol for analyzing these studies. As is the Pancreas talk, I'll show you there were a number of diabetes studies that were evaluating whose major outcomes were either major adverse cardiovascular events or a hemoglobin A1C target. And they basically executed the study using the new cohort method. And what you can see in this table is they really nicely showed that they could replicate the RCT findings in real time using the database approach with similar relative risks and estimates for reduction with these different medications. And this nicely showed that with the statistical complexity and the portability of analytical tools, we can really replicate a lot of phase 4 studies where we understand the impact of medications after their approval. Next, I just wanted to highlight the power of a common data model even on retrospective work. A lot of work that trainees do and a lot of work that clinicians do will be retrospective cohort studies where data is collected from the EHR. The process is often very time-consuming and difficult, but a group of Korean hospitals got together and linked their EHR to the OMOP common data model, and then were able to look at subsets of data and do different analytical tools. In this case, they were evaluating the question, do hospitalized patients with a high triglyceride level have more severe pancreatitis than a low triglyceride level? And they defined severity using lab values such as CRP, creatinine, as well as ICU admission. In this case, they used an OHDSI open source tool called Atlas. This allowed them to evaluate if this cohort 1 was feasible, create the cohort, and conduct descriptive analysis. And they then were able to output this data to perform simple linear regression and make these comparisons. This is their graphical abstract. The really cool thing here is they took something that is really time intensive and made it relatively simple using an automated extraction from the EHR and open source simple tools that they did not have to recode or re-engineer. What you can see in their findings in this graphical abstract is they found that the CRP as well as the rates of ICU admission were higher in the high triglyceride arm compared to the low triglyceride arm, showing that the triglyceride level may modulate severity of acute pancreatitis. The last example I'll give you, just to show kind of what is potentially on the horizon, is whether we can use open source tools to predict who's going to develop pancreatic ductal adenocarcinoma, given the poor outcomes currently, with patients who present symptomatically. This was a really nice paper in Nature Medicine, and what they did is they used an open source R machine learning tool, and they fit a machine learning model to predict PDAC with data at least 6 months prior to developing pancreatic ductal adenocarcinoma in a data set of 6 million patients in Denmark. They then validated it with 3 million patients to the Veterans Administration's clinical data warehouse, both of which use the common data model. The first figure in panel A you can see is the different models, and they were comparing their area under the curve or their accuracy. And you can see, the transformer model was the best model, but all of these were significantly better than just a model that tried to randomly guess. They then took these models and asked, what's the prediction interval? Could they predict even 5 years out? And showed an area under the curve of 0.8, even predicting 5 years out, which is good to great for its accuracy, and demonstrate the promise of this tool. One nice thing is they made their model widely available. It is open source, called Cancer RiskNet, so there's a lot of work on refining the model to see if we can do a better job of predicting pancreatic cancer with just variables present in the EHR. With that, I'd like to conclude, and I hope I've given you a sense that the large-scale clinical data mining can fill an unmet need in the clinical research landscape, providing pharmacovigilance, pharmacoepidemiology, and understanding treatment utilization. The real game changer here has been the Common Data Dictionary, or OMOP, which facilitates the easy conduct of analyses across data sets without having to reformat code and analytical tools. The real game changer here will be if that EHR platforms embrace common data models, particularly things like Epic Cosmos, which has a wealth of data, making retrospective analyses much easier. There's also an evolving set of open source analytical tools using R and Python that make things like machine learning considerably easier to perform. And really, machine learning has the potential to revolutionize disease detection, prediction, and treatment assessments. And there's a lot of interest in using large language models because the main kind of bottleneck to date has been the unavailability of large data for these kinds of models. So a lot of this, is very promising that this can potentially change the field of pancreatology using large data sets. With that, I'd like to thank you all, and we'll take questions at the end.
Thank you very much for staying on time, Nikhil. In the interest of time, we're going to go through all the talks first, and then we're going to have at the end a discussion panel. So hold off with all your questions. So the next speaker is Kyle Galton. Actually, he's a local hero. He is here from UCSD, and he's going to give us an update on PancBase.
Okay, well, thank you so much for the invitation to speak. I come from the diabetes world, and I'm going to talk today about a new resource that we're funded to develop called PancBase, which is an effort to provide knowledge of the human pancreas to understand diabetes, but we're also interested in expanding, the knowledge that can be leveraged from this platform to understand pancreatitis and other pancreatic diseases, as well as understand the connections between diabetes and pancreatic disease. And I'm speaking on behalf of our large team, and I'll introduce them in a minute. So there's an amazing opportunity, and kind of the genesis of this project is the incredible wealth of data that's been generated in the human pancreas and continues to be generated. And this is spread across many different repositories, so repositories that generate data on human islets or human pancreas, clinical resources as well. There's also a wealth of different types of data that are being generated in the pancreas. So genomic data, genetic data, other types of data, functional information, electrophysiology, and so this incredible wealth of data being collected on human islets and human pancreatic donors, again, is a real opportunity for us to improve our understanding of diabetes and other pancreatic diseases. But this opportunity comes with some challenges as well. So what are some of the barriers to utilizing data in the pancreas? So one is that the standardization of data and metadata is given that these data are spread across many resources, requires harmonization. They're dispersed across many locations. They are described in different ways. They may have different metadata and ways that the information is collected. And so we need to work towards standardizing and collecting all this information and representing it in a uniform way. Two, there's like I would say a lack of availability of high-value resources derived from data that's harmonized across these different resources, and there's few harmonized resources that span data being generated across different pancreatic resources, for example, single-cell maps and so forth. And there's often unclear provenance and reporting of data and biases and outliers in these resources that needs to be represented in these resources to make them more useful, particularly for machine learning and AI type applications. Third, there are limited interfaces to interact with these data that are useful to a wide range of researchers, from computational biologists that want to utilize these data in model training and evaluation, as well as basic researchers who want to look up genes of interest or develop hypotheses for pursuing mechanistic studies. So the goal of PancBase is to address some of these challenges, to collect, harmonize, and represent pancreatic data back to the research community in a variety of ways. And so we have a resource at pancbase.org. It's in beta phase right now. The initial version of this database was released about a year ago. And so what this resource represents is a comprehensive repository of knowledge, data, and metadata of the human pancreas. A lot of effort we spent on this project to create harmonized resources that are tools that can be utilized by researchers and represented in the platform. development of different interfaces through which people can access these data and query it and utilize it in various applications, again, spanning basic biologists to more advanced, computational biology, as well as thinking about how we can implement machine learning, AI-based tools to learn from these data to again, improve the representation of the data, but then also gain deeper insights into what it can tell us about disease. So we have a large investigator team, and these investigators are spread across many institutions, some here in California, some on the East Coast, and it's a diverse team that spans individuals who have expertise in development of knowledge bases, people who are experts in computational biology, also people who are experts in pancreatic biology. And I should note as well that this is an initiative supported by a consortium called HERN, which is the Human Islet Research Network. And the goals of HERN are really to understand the pathogenesis of type 1 diabetes. But again, the remit of this project, I think, goes beyond just type 1 diabetes to understand generally pancreatic disease, diabetes, pancreatitis, and other forms of pancreatic disease, given the connections between these different diseases of the pancreas. So I'll talk a little bit about the key products that we're working on making as part of PancBase and how we can then utilize them to gain deeper insights from data. So as I mentioned a couple slides ago, we spent a lot of effort creating harmonized resources that span data and metadata that exist across different resources. And so examples of this are single-cell maps that collect data across different repositories, bulk RNA-seq, other types of genomic data has been a big focus thus far, as well as harmonizing metadata that exist on donors and biosamples of the pancreas. And if you look across all these resources together, there's about 3,800 human donors that have some level of information that's been collected on them across all these resources together. So the number of individuals that have some form of data is actually quite vast, and so we collect that information as well. And I'm highlighting specifically on this slide a single-cell map, which collects data from a variety of pancreatic resources and 140 islet donors in total, and we have representation of different cell types in the pancreas. And this map can be utilized to ask questions about gene expression levels of genes of interest, changes in gene expression in disease, and so forth. So what's the generation of these resources, and then what do we do with them in the resource that's being created? So on the left-hand side, the generation of these resources consists of standardization of the pipelines used to process data, quality control, aligning metadata describing the donors that are comprised of this map, as well as a lot of effort to validate donor identity and ensure the fidelity of the donors and the data being fed into this map. On the right, what do we use this information for? So first, just provide these resources for people to download and use. And so we have web portals, APIs that allow people to access these through web, direct download through the web or programmatically through APIs. Tools that visualize queries that users can perform to search genes of interest or changes in a gene and disease. More like AI-based representations of data, so knowledge graphs that encode connections between genes and the cell types and the disease states that they represent, that you can sit AI tools on top of to learn from. And in gray on the bottom, something that doesn't exist yet in the platform but is in progress is developing web-based notebooks and tools that researchers can go to and load these maps and apply their own tools or load, maybe data that they've generated themselves, and make it easier for people to, tinker with analyses or rerun things in different ways, so that they can, perform the analyses they're interested in or modify things the way they want or incorporate additional data into these existing resources. Okay, so I'll briefly touch on each of these access mechanisms to understand data that we're creating from these harmonized resources. So one is a database that hosts all the underlying data. And this contains both the resources themselves, so like single-cell maps, donor metadata tables, as well as external resources of high value, so data from resources like GTEx or like pathway gene sets or GWAS data that's commonly used to interface with the resources we're creating, as well as the raw components, so like the individual process files, individual metadata records collected on donors, as well as extensive documentation about the standards that were used to collect all this information, the workflows used to create it, and so forth. So all this information is available in this database. And again, There are APIs, web portals, and there are even direct access to buckets on AWS, basically a variety of ways that make it easier for people to be able to access this data. The second way we provide this information is through web-based interfaces that visualize these resources, and in this case, a single-cell map, and allow people to query it to ask specific questions of interest. So in this case here, you could search for a gene of interest, and in this case, let's search for trypsinogen, PRSS2, and you can see where this gene is expressed across the single-cell maps. It's SNR specific gene, of course. The expression level of trypsinogen across different cell types in the bottom right, and not shown here, but you can also facet based on disease state, age, BMI, other donor metadata information to understand changes in the expression of this query gene across metadata that the project is collecting. Another interface to these resources that we're collecting, as I mentioned earlier, is a knowledge graph, which encodes relationships between different entities. And in this case, I've searched for gene HLA-A, which is the class 1 MHC, which is upregulated in type 1 diabetes. This knowledge graph summarizes information about this gene, what's known about this gene in the literature in terms of its role in pancreatic disease, as well as connections between this gene and, say, interacting partners and other relationships that are being fed in, as well as an AI-based summary of all this information. So you don't have to do all the connections yourself, but it can produce a high-level summary that tells you key pieces of information about the gene you're interested in. We also connect different resources that the project's collecting. So for example, if we take the single-cell RNA-seq map and then all the donor metadata that we've collected, we can put these two together to ask whether there are changes in gene expression in disease, and there's different visualizations, for example, like differential expression in type 1 diabetes or other facets of the donor metadata that's collected. And we have hundreds of donor metadata fields, and so there's a variety of ways that these data can be accessed to look for changes across a variety of phenotypes. Okay, there's also a quite extensive engagement and outreach effort as part of this project, and that I think comprises two things. So first, engagement with the resources and data providers directly to understand the data that they generate, understand provenance, understand the metadata they're collecting, so we can do a better job of representing all this information back to the community, as well as improve our harmonization of this information across different resources. Also leverage the insight that the generators of these data have to think about the types of questions that are ideally asked from these data, as well as the outreach to the broader research community, just like today, to understand community needs, data analysis standards, tailored development of the platform to address questions that the research community is actually interested in, as well as hopefully facilitating user contribution of data, tools they're developing, analyses they're interested in, ideas back into the platform. There are also opportunity funds, so funding opportunities provided by the NADDK through PancBase that investigators can apply to. And if you go to the website, you can find more information about that. If people are interested in performing analyses or contributing data to the program, there are funding mechanisms that facilitate that. Okay. What are we doing now, and what are the areas that we're moving into in the upcoming years? So one is expanding the data types that are represented in the platform. To date, a lot of the information that we've collected is genomics, gene expression, regulatory element activity, genomic type data, but as I mentioned at the beginning, there's all sorts of different data types collected in the pancreas, functional data, spatial data, proteomics, genetic data, et cetera. So expanding the representation of data, as well as the resources that we derive from these data and their representation in the platform. Two, there's been a lot of interest and effort to try to develop agentic AI interfaces, and so that I mean like AI agents that are able to query the platform and then summarize back to users information that's present in the platform, so that helps both with navigation through these platforms, as well as help individuals summarize information that the platform contains to help them develop hypotheses or understand where they might get additional information and so forth. So a lot of work to try to sit AI-based agents on top of the platform to help guide the user experience. And I mentioned the analytical library to allow people to perform their analyses and provide code to make it easier for people to do analyses they're interested in themselves. and then implementing use cases, particularly those that utilize machine learning and AI. So lastly, I have just some considerations for the community here, for APA, and how we might work together in terms of building a platform that not just covers diabetes, but also pancreatitis and pancreatic cancer and other relevant diseases of the pancreas. So things to think through are what are the high-value data sets and resources for pancreatic disease that have been generated in the human pancreas that can be aggregated in this platform? What are the unique aspects of representing pancreatic disease, say, relative to diabetes that we should represent in donor and data set metadata? Like a lot of our focus on harmonizing metadata to date has been specifically around diabetes and type 1 diabetes and staging of type 1 diabetes, and surely there's considerations for pancreatitis in the same vein that we can improve upon. What are the key open questions about pancreatic disease that we can answer using these data? And then also, who are the investigators consortia that would be willing partners to work with PancBase to try to improve the representation of these relevant data in the platform? So if you're interested in participating or want to know more, so we have a community forum where you can submit questions or get in contact with the team. There's also a help email that you can email us about, and there's many ways to get involved in the project. So applying for funding opportunities, like I mentioned, contributing data sets, helping to develop standards, suggest use cases. We have a lot of collaborations in the diabetes realm in each of these areas, and I think it would be an opportunity to do this here as well. Okay, so thank you so much, and I guess I'll save the questions for the panel. Thank you.
Thank you, Dr. Galton. And our next speaker is Dr. Kales from Northwestern University. She will talk about MRI-based AI imaging for chronic pancreatitis and pancreatogenic diabetes.
Thank you so much. Thank you for having me. I'm a physician scientist, and I'm going to explain what we are doing in our lab. Okay, let me introduce you briefly our lab, Machine and Hybrid Intelligence Lab, and we are strategically located in downtown Chicago, and our PI, Dr. Ulaş Bağcı, has a dual appointment, both medical school and also engineering school, and we are part of the AI Center and also Cancer Center, and we have more than 30 members in our lab, and we have a great computational sources to run the AI experiments. And we are happy that because our projects are supported by NIH with the several grants, and as you've seen in here, we are dealing with the different medical problems in medical imaging, and also if you look at carefully the titles of our projects, we always keep explainability and interpretability and the human-in-the-loop systems in our medical problems and our projects. And we are, in our research, spans with different size of medical imaging and different AI problems, classification, prediction, federal learning, and privacy-preserving learning, and multimodal learning, and foundational models. So trust in medical AI is really important, and there are 3 steps in those 3 pillars in the center of in our research. The one is algorithmic robustness, we should be always ensuring the algorithms could be working in across diverse data sets, and interpretability and explainability. We should tell clinician how AI could decide while decision making, and human-in-the-loop and expert-in-the-loop systems. We could integrate the expert's knowledge into the AI models. And AI field is moving so fast, and every day new architecture and the models come into play. And when we look at the whole timeline, in 2000s, those are more feature-centric models. In 2010s, deep learning got popularity. Then now, foundational models. Medical imaging is still behind of these foundational models, which the other disciplines got some acceleration, but we are going forward. So let's imagine a world we identify chronic pancreatitis at its early stages before irreversible fibrosis, diabetes, and exocrine insufficiency develop. And a world where AI algorithms could analyze all imaging data and the clinical data overnight, which we as a human could analyze all data kind of decades. So this is the healthy pancreas, and with AI we can predict the future with the clinical data and what's going to happen in an imaging side and also clinical side for this patient. So let's get back into reality. What have been doing in our lab so far? We are working with the pancreas segmentation. You all know very well pancreas is really hard organ, even to differentiate the borders from the different organs in the scannings. And we did also pancreas part segmentation and cysts and tumor segmentation. And fat is also very popular, tissue segmentation. And we make all our data open source, and we make all our model publicly available, and we also developed a user interface which researchers want to use, and it can automatically extract the pancreas, and it can segment the pancreas and gives you the volume with a 3D version. And we use also AI models to predict the diagnosis and predict the prognosis, and just for a diagnosis, it's for all disease, pancreatitis, diabetes, acute and chronic pancreatitis, and also diabetes and pancreatic cancer and IPMN. And we also use federated learning because we know, we witnessed the data sharing is really hard. And AI could be used for different projects and different medical problems, but major approach could be categorized under these three titles, like standardization and harmonization, because it should be always intensity correction and data homogeneity. Otherwise, AI could detect where those images come from, like from different vendors, instead of focusing on the diagnosis and the subtle changes. And segmentation, we should always segment the pancreas when we go deeper, like what's going on as a subtle change in the tissue level, and also prediction algorithms. And outcome is really important. We could hyper-tune our model based on the outcome and what we're gonna see and what's our expectation from the AI models. Let's think a patient. Patient got a scan, then later we can narrow down T1 weighted and T2 weighted scan. Then later we segmented the pancreas with our algorithms, which called is PanSegnet. I'm gonna go in details. And with those segmentation, we can fuse the models, radiomics and deep learning. Then we can give the chronic pancreatitis severity or what's gonna happen in the future and what are the expecting risk for these patients. And this is the harmonization. Actually, this is nothing new, but we should do always before the segmentation. And pancreas segmentation historically is very challenging due to the nature and the boundaries of the organ. And also volumetry is really important. In our daily settings, it's not easy to calculate from slice by slice and always asking the radiologist because it's not easy calculatable. And also segmented pancreas could be helpful for autodiagnosis and the disease prediction and the prognosis prediction. And we give the volumetry, and we also segment the cysts and extract from the volumetry, and we can do the classification. Classification could be based whole scan versus pancreas region. When we narrow down in the pancreas region, we have a better accuracy in the predictions. And this is the model and the data which we worked on. This is the state-of-the-art model and also the first model in large-scale data, both using CT and MRI. And we did this 1,400 MRI, and we made all public, and we also made public our model. Even we created a user interface. You can scan the code and go to the paper, and then you can go to the model and GitHub page. And this is the first ever multi-site, multi-MRI pancreas segmentation model. Both worked in T1 and T2 with a high accuracy. We use DICE score as an evaluation metric. If DICE score means AI model could predict the whole organ in a whole volume, which it can predict whole volume, the DICE score would be 100% or like 1. And now the current model in our algorithm, we can predict T1 and T2 over 85 DICE, and also in CT, it's always better. And you may see some visual results, and the green contours the ground truth, which made by the human radiologist, and the red is the AI predictions. And we have over 90 DICE accuracy in the CT, and both T1 and T2, as you may know, like all the vendors have different acquisition protocol, and our model harmonized everything before segmented, and also those are the model. We have a good accuracy, both T1 and T2, over 80, just one click, just with the AI. And we shared all data. You may go to data. It's all annotated, and you may use while develop your model, and you may do another experiments. And this is the one-click user interface. I also added the QR code. It will get you through the page, and you may download your computer, and when you upload just CT and T1 and T2, it gives you automatically the pancreas segmentation, and also it gives you the volume, and you may also download as a different file. And we also tried our model, even we have never trained the model on out-of-phase MRI. We have a good DICE score over 87. And we also test the algorithmic robustness in the pediatric population, and without any training. We did the segmentation on T2 images for healthy and acute pancreatitis and the chronic pancreatitis, and we have a really good results. And as you may see in here, in all, we have 85, and healthy children 88, and acute and chronic, it's around 80. And if we look at the other evaluation metrics, it's all clinically compatible. So it shows our algorithm is robust. It's also published, and we also share the pediatric data with all annotations. And this is, oh, sorry. This is the also link for the whole data set. We also segmented the pancreas by parts because each part has an importance in the disease and the etiology, and we take a reference point, superior mesenteric man, and like the medial, we segment it as a head, and the body and tail, since there is no correct definition, we split into two parts, and we have a good accuracy in both the part segmentation. Sorry, yeah, those are the visual results and the models which we have used. Those are all deep learning models combined with the transformers. And we also can segment tumor and cyst, because you all know very well, like cyst can progress into cancer, and we do scan and surgery. And we also generate our data because we all witness data sharing is a really big problem. And we do risk stratification. I'm going through the IPMM because the pipeline is similar for all the disease. And we expanded our data set, and those are the historical papers our group worked on. And from 2019 to 23, what changed? We extended the data set, and we tried the transformers instead of deep learning algorithm, and we integrated radiomics. And those are the results. You see the AUC, which is with deep learning, it's even maximum 80. We should have better AUC, and we integrated radiomics. Radiomics is they are interpreting the image into numbers, and they are extracting the features, and we integrated the features into the model, and then we compare the model performance versus normal low grade, normal versus low grade, and normal versus high grade, and it's helping to risk stratification. And After many experiments, we have reached out more than 80% and a better AUC, both in test and validation set. It can also fail, so it should be human in the loop, and we should know why it's failing. And this is one of the explainability model, shows us the where AI look at and how make a decision. It's called the information bottleneck algorithm, and it will give the clinician the reasoning. We can also stratify the acute pancreatitis based on the CT, and we use the machine learning model, and we have a good accuracy AUC, and also we shared this data. We also segmented the edema, and we used radiomics techniques, and it's all published, and we combined the models with machine learning and also transformers. And we recently got CP-DPC data. It's still ongoing research, and we are segmenting the pancreas by parts and classify based on the Cambridge criteria, which it's still going on discussion, which would be the better classification method. And we, even without deep learning, we have more than 74 AUC, and we could combine the transforms and the deep learning into the model. So AI is rapidly transforming the medicine, and gastroenterology is the leading with the FDI and other approved applications, but we have still challenges, and we need to come over those challenges with the multimodal data and more data, labeled data, and increasing the heterogeneity and generalizability. So privacy and HIPAA got a big share in our data sharing. So we proposed the federated learning model. There's a central server, and we don't need to share data, and each center can run their algorithm, and the model can be updated by itself, and it gives the results. We simulated with the 7 centers data, and we have 70% DICE score. It's just without sharing data. And we also did the part segmentation with federated learning, and we have also good results, clinically usable. And FET is also a hot topic, and there are some FET segmentation algorithms, but which is worked on Dixon and also CT. In Dixon, you may do threshold, but not all the patients have the Dixon, so T1 and T2 is the most used sequences, and we make an experiment to segment visceral adipose tissue and the subcutaneous adipose tissue and intramuscular fat. And we have currently some challenge and trying to overcome those challenges with the model development, and models still fail, and you may see even there is no segmentation, and this is the CT model. And so we didn't solve all challenges. We still need more data, heterogeneity, and we should overcome the data sharing issue with federated learning, and we should always have the results generalizability and interpretability, and it could be whole pipeline to see the pancreatic disease where going in direction. And it should be always multimodal deep learning, explainable, and federated learning should be on the table. And CP is a ticking bomb, and we can intervene with the AI, but when we intervene with AI, we should discuss what will we do, and how can we make it faster. Thank you so much, and those are our collaborators during the SysTex project, which they made possible with us sharing those data and developing the model. Thank you so much.
All right. Thank you very much. And in the interest of time, let's continue with the last but not least speaker, Dr. Ravi Vairavelu from University of Pittsburgh. He's going to talk to us about using PVOS approach for drug-induced pancreatitis injury.
All right, thank you very much to the organizers for inviting me to speak here today. Sorry, I'm just going to adjust so I can see the better. To start off, I have no financial conflicts of interest to disclose. The research that I'm presenting here was supported by those 2 grants from the NIH, and I work for the Department of Veterans Affairs, and these views are my own. This talk is going to unfold in 3 parts. The first is I'm going to review the current evidence base for drug-induced pancreatitis. Following that, I'll describe a method that our group has developed to study drug-induced pancreatitis using large real-world health databases. And finally, I'll present some selected results from that. This has been about a decade-long project, and I would be remiss not to mention our multidisciplinary, multi-institutional team who have been instrumental in having this research completed. For this audience, I don't have to explain what the most common etiologies of acute pancreatitis are, but I will point out that clinically, for non-gallstone, non-alcohol pancreatitis, although there are several potential etiologies, we often label these as idiopathic in the clinical setting. At the same time, we know that for decades, there has been increasing incidence of acute pancreatitis across the entire world. At the same time, the prevalence of polypharmacy and medication use has also been increasing. It would be very naive to suggest that these are 100% related, but because of the striking mirroring of these 2 graphs, it induced our group to study this further. And the literature is overwhelming to review. From the past 20 years, over 850 medications have been reported to the FDA as potential causes of acute pancreatitis. A list of some of them is here. At the same time, a group from Maimonides Medical Center in New York City has been categorizing these reports based on their strength of evidence. And you can see very few medications have had randomized control trial evidence. Slightly more, only 14 have been studied in pharmacoepidemiology studies such as case control studies or cohort studies, and then the rest have been case reports of varying quality. Our goal for this study was to solidify the evidence base for the pharmacoepidemiology studies and improve it for case reports. And so we came to this overwhelming problem of how are we going to study 850 medications at one time. After some reflection, we gained some inspiration from genome-wide association studies, which had been doing this type of work for decades. And so I know many of you in the audience do these genome-wide association studies, so I'm going to ask you in advance to forgive my epidemiologist simplification, but I'm going to describe a hypothetical acute pancreatitis GWAS so that we can use it as an analogy for how we would do our pharmacoepidemiology study. So to do this GWAS, what we would do is identify a group of patients, identify those who had acute pancreatitis, and separate the patients with acute pancreatitis and call them cases from the patients who didn't have acute pancreatitis, who would be called controls. We then genotype these 2 groups of patients for tens of thousands of single nucleotide polymorphisms, which represent differences in the ways that the genes are encoded. For each SNP, we would then calculate the prevalence among cases and controls and compare them. We can summarize this comparison as an odds ratio and calculate the statistical significance using chi-square testing to get a p-value. If we then map all of these chromosomes and associations, if we map all these SNPs to chromosomes and the associations, we can develop a Manhattan plot, which is shown at the bottom right corner, which describes the chromosome on the x-axis. The y-axis is the negative log of the p-value, so higher values represent more statistically significant results, and the dashed horizontal line represents the adjustment for multiple hypothesis testing. The dots above the horizontal line indicate medications that are potentially SNPs that are potentially associated with acute pancreatitis. And indeed, this type of study has been used to replicate pancreatitis susceptibility genes such as CTRC, SPINK1, and PRSS. So now we'll use that as an analogy for our pharmacopoeia-wide association study, or PharmWAS. Again, we would separate our patients into people who had experienced acute pancreatitis and people who hadn't. And now instead of genotyping them, we're going to interrogate their medical record to identify what medications they were using around the time of acute pancreatitis. Since the controls did not experience acute pancreatitis, we give them an index state that matches with a case who had similar demographic features to them. We call that the index date. And so now, for each medication that we want to study, we're going to calculate the prevalence of utilization around the index date for each medication and compare it from cases and controls. Again, we can summarize that with an odds ratio and get p-values, and we will plot it on a volcano plot where the x-axis represents the odds ratio, values to the right represent medications that are potentially associated, and values to the left represent medications that are inversely associated. The horizontal line, again, is the line of multiple hypothesis testing, and so the associations above that are potentially statistically significant. So this is easier said than done because we have the possibility of having false positives. So this is a study from another group that had attempted a similar methodology to study myocardial infarction. They studied about 120 medications and found that almost 20% of them were associated with myocardial infarction, which isn't clinically reasonable. And so the study suffered from many false positives. So before we could implement our methodology, we had to create a pipeline of analytic methods that implemented pharmacoepidemiology best practices. I'm going to highlight 3 of the 7 here. Number 3 was that we were using a case control study design to match on age, sex, and pancreatitis risk factors such as history of alcohol use disorder to isolate the medication effects. Number 4, we're focusing only on new users of each medication because new users are more susceptible to adverse drug effects compared to people who've been maintained on a medication for a long time. And then number 6 is that we control for confounding by indication using high-dimensional propensity scores. So this allows us to isolate the effect of the medication from the effect of the reason why the medication was prescribed. And then after implementing this pipeline, we needed to validate that this methodology could work to reproduce associations that are already known. So these are 2 validation studies that we published that reproduced known medication effects for C. difficile infection and gastrointestinal bleeding. These were performed in 2 different studies from 2 different countries, 2 different databases from 2 different countries in over 1 million patients collectively. Having done that validation, we then felt comfortable trying to study drug-induced pancreatitis. So a few years ago, we initiated this study using data from the U.S. Veterans Health Administration. The data span over 2 decades. We have identified almost 30,000 cases who experienced first-time acute pancreatitis in the VA, and the controls are about 4 controls for each case. Overall, there are about 600 medications that have enough utilization for us to study. So here are some selected results. I would describe these as signals that still need to be validated. So this first table represents medications that had already had a clinical suspicion for being associated with drug-induced pancreatitis, similar to what I had shown before in the pyramid. The middle column represents the odds ratio for if we are looking back from the index date back a whole year for when the medication was started. And then the last column represents the same, except we're only looking back 90 days. So we're seeing very strong associations with azathioprine and 6-mercaptopurine, and also some strong associations with other medications that had been suspected, such as furosemide and sulfamethoxazole trimethoprim. But as described these only as signals, one thing that could help us with some biologic plausibility is to see if these medications had consistent time from the time that the medication was initiated to when the patient experienced acute pancreatitis. That could be indicative of like a biologic mechanism such as a type 4 hypersensitivity reaction. And so here I'm overlaying plots that describe when the medication was initiated relative to the index date, which is all the way on the far right of the slide. And we can see, most striking for azathioprine and 6-MP, again, medications that have randomized control trial data for their association with drug-induced pancreatitis, that it's about 50 days from medication initiation to acute pancreatitis. And I also note that the curves are flatter for the other medications, indicating that we are not seeing that same type of biologic plausibility for those associations. Here's some list of anti-infective medications. We're seeing a very strong association for tetracycline and metronidazole, also some for clarithromycin. When we overlay the temporality plots, we're seeing good temporality for tetracycline, metronidazole, and fanquinone, which is an antiparasitic medication. And these are a group of medications I call oddballs because they have either a unique mechanism of action or a unique indication. And the striking associations are for coal tar, acetylcysteine, and hydralazine, and they're also showing some consistent temporality. Again, I would only describe these as signals. We are doing ongoing analyses to validate these findings using the Bradford-Hill criteria, which has guided us with a framework to conduct other studies such as re-exposure analyses, dose response, and reproducibility in a second database to identify which of these are true associations that have clinical implications. And like the other investigators here, our goal is to disseminate these findings in a way that is interpretable and usable for both researchers and clinicians. We are designing a web app that will show all of these 7 different analyses in 2 different databases for 600 plus medications. We would be very grateful if people here are interested in user testing this app so that we can disseminate our results in a way that's effective for clinicians. Well, thank you for your interest, and I look forward to your questions.
All right. Thank you very much, especially for being on time. With that, we would like to open the floor for questions from the audience. So please step forward. Don't be afraid. I know the microphone is rather close to the front. Sorry, you get your steps in, so yeah.
There you go. Thank you very much. Suhail Hussain, I really enjoyed the talks. I had just 2 brief questions, one for Kyle. Thanks for coming back, and I hope to see you more often. PancBase is such an important resource for the endocrine community. I know there's, as you mentioned, an interest in incorporating it within, or incorporating exocrine tissue or pancreatitis and things like that. Can you give us more of a timeline? We have open source or data sets that if you would like to curate, we would certainly be very happy to give to you. The second is for the last speaker, really nice talk. I guess I was a little surprised, and I think this is sort of early work, so I'm really interested in understanding how you're validating it, is that asparaginase is so highly linked to pancreatitis. The latency period is usually much later, and on the other hand, things like retinol are protective, at least in many studies. So you're picking up a phenomenon that maybe aren't exactly what I would have expected, and I just wonder about the validation.
Hello? Okay. For the question about PancBase, so the timing could be now or as soon as you want, and I think if we're able to connect on where those data are and put people together to start integrating it into what we have, I think that could happen soon. But we have, so there's a lot of like purified islet data, and like a lot of what I showed was purified islets. There's also a fair amount of whole pancreas data, like genomic data and other data that we have not incorporated yet, but would expand the repertoire of like maybe cells and the depth of cells relevant to pancreatitis that we're able to study relative to what is in the purified islets. But yeah, I mean, if there are data sets you know of and that would be useful for this program, I think that's something that could happen immediately.
Yeah, thank you for your interest. I would just describe the work as signals for right now. We were seeing a signal for asparaginase. It did not have the same temporality as we saw with the other, especially some of the other stronger medications, but we will be continuing to assess the biologic plausibility in different aspects, at least 7 different analyses that we have planned, including a second database. So there's still very much an opportunity that the signals that you expect to see will show up.
Thank you very much. Just a reminder, sorry, we have 6 minutes left, so please
Okay.
keep it brief. Thank you.
Okay, fine. Thank you. Thank you very much, panel, for all the presentations. Very enjoyable. I just wanted to address a couple of comments and maybe a question or two to Ravi. Make it very brief. A very innovative approach and very sensible to describe as signals. I suppose one of the things about drugs and acute pancreatitis is that the latency period may not just relate to the drug, but it may relate to other factors, that whether there's an alcohol, whether there's a fatty meal, what have you, what precipitants there might be. Capturing all of that is very difficult. And then the odds ratios that you come out with may not necessarily be indicative of the likelihood itself, because it's dependent on the data sources you have and how well documented they are and so on. The other thing is that there are other sources of data to assess biological plausibility. So hydrochlorothiazide, for example, there is a very good 1960s New England Journal of paper about chlorothiazide, and it was tested in mice, and it demonstrates how likelihood, and I think those studies should also be incorporated. But nice work, thank you.
Yeah, thank you very much.
Thank you for some fascinating presentations. So I'm Rishi Mukherjee. I'm a scientist, clinician scientist working on AI/ML approaches in translational drug discovery for acute pancreatitis in Liverpool. I was just wondering, with regards the opinions from the panel about the need for metrics on hallucination for all these AI/ML models, particularly considering translation to clinical grade models and the increasing requirement from the regulatory authorities that in the next few years this will be important as a comparator and basically compared to all standard clinical approaches to allow us to assess how good these models are. So open to the panel.
Actually, the studies which we have done with the imaging model we don't use the LLMs. LLM has a potential for the hallucination, and but deep learning model and transformers with the imaging data, there is no possibility for the hallucination. But when we use the LLMs, we should be always careful, and we shouldn't make them as a source of truth. And regulatory side, FDA and the others, even there are also some models, they can replicate the lab studies, but as long as FDA still accept the conventional methods, I don't think there will be a reason for the approval parts.
Thank you.
All right, it's 12:30, it's lunchtime, so I will thank you for everybody joining us. Enjoy the lunch, thank you.